what a wonderful start to this important symposium. You know, I sit here and I look out at the audience and so many familiar faces and so much hard work that's gone into developing this field of dry eye. You know, it's so important for patients, for physicians, for sponsors, for researchers, as we've heard today. So we've talked a lot about the unmet medical needs this morning and all the different influential factors. And so today, at this point, we're going to talk about what is in the future. What are the therapies that are currently being worked on? So I'm happy to start with a history of the existing U.S. approvals, product approvals. So as we know, it started all with Restasis in 2003, 20 years ago, if you can believe that, 21 years. And this is the first product that came to market. It is approved, as you know, for tear production as measured by the Schirmer responder rate. And it's effective at six months after treatment. Then it was a solid 13 years till the next product came to market, and that was Xiidra. And Xiidra was the first that actually addresses both signs and symptoms, inferior corneal staining as well as ocular dryness. And then shortly after that, we had our first tear producer or tear stimulant product, and that was a medical device, TrueTear. And then in 2018, we saw Cequa, which is another version of cyclosporin with a unique vehicle that helped with the onset of efficacy. Then in 2020, our first soft steroid for the treatment of dry eye and flares associated with dry eye came to market, Isovis. And then in 21, Tyrvaya came to us, and this is a product that has the same tear stimulant, but through a chemical stimulation. And then exciting, in last year, 2023, we saw 2 new products come to market from our friends at Novaliq and now marketed by Bausch & Lomb, is the Mibo Tear. And this is the first product to target evaporative dry eye for both signs and symptoms, and then Vivai with the treatment of dry eye with cyclosporin and their unique delivery mechanism as well. As we know, there are some challenges with some of the products that are currently in the market. We see that maybe there's a strong focus on signs, not as much on symptoms, or there may be some adverse events or some longer onset of action. So at this point, we're going to talk about the future and what are some of the exciting products that are being developed right now. All right, so I'm going to start with the first presenter. This is Eric Schlumpf from Stuart Therapeutics. And we're happy to have him talk about his very unique technology, ST100, that is targeting the neural processes on the ocular surface, both the cornea as well as the conjunctiva, and how this may potentially address one of the underlying etiologies of dry eye.
Thanks, George. Good to see you.
Nice to see you too.
Yeah. Good afternoon, folks. It's great to be here and great to see all of you. How many of you were very excited to be reminded about how old you were this morning, first thing? When you look on our website, we definitely average out to boomer. So that says something about us. But we've also got somebody who wasn't on the chart. Our chairman is kind of a neo greatest generation guy. So we're pretty proud of Bob. And he couldn't make it this time because he just had eye surgery. But thrilled that we're a part of this and great to be associated with OIS and Aura. I want to talk about visacomatide, which is also ST100, which is our comprehensive therapeutic for dry eye disease. We're a late clinical stage company. The peptide that we're developing is a synthesized polypeptide that mimics a fractional strand of human collagen. The way it works is it binds to disease-damaged collagen. That binding provides a number of features that we'll talk about here in just a few minutes. And we had a pretty interesting journey as we went to our phase 2 trial and then digging even deeper into the mechanism of action I'll share with you today. Collagen is a biologically active and very important component of the eye and the optic nerve system in the human body. It is a cell signal ligand binding site for important proteins. It helps the microenvironment stay healthy in homeostasis. When we repair damaged collagen, we help restore that homeostatic role for collagen. And collagen damage is pathogenic in a number of ophthalmic indications, including dry eye, Sjogren's, neurotrophic keratitis, et cetera. The first experiment that we ran, the first study we ran with the College of Mimetic Peptides, or CMPs, was a lamellar keratectomy, where we removed the epithelial layer and some of the stroma edge to edge on mouse eyes. And we put a very low concentration, single drop of the peptide on these animals just to see what would happen. We had really no idea. This was our first foray into this. And what we found was that, sure enough, in 16 hours, we had full recovery of the corneal epithelium, full adherence to the to a basement membrane and the stroma, and significant reduction in inflammation signs in the stromal tissues. So, hey, something that deals with one of those things we talked about earlier today, which is corneal ocular surface tissue health. Very good thing. We thought we got there. So let's go crazy and let's do a phase 2 clinical trial, which we did. So the ST100 phase 2 trial addressed visual function, symptoms, staining, and lacrimal tear production within 28 days. We hit a visual acuity endpoint with significance on the second and third day. We had symptom improvement by the second week, staining by the week 4, and lacrimal tear function by week 4 as well. The drug is very well tolerated. The safety profile is very nice. The drop comfort scores were similar to typical artificial tear products that patients are using today, like Sustain. When we looked at all those endpoints and the way that all came together in that trial, we said to ourselves, okay, we think what we're having here is an impact on the lacrimal function overall. So let's take a look at a couple of different animal studies that'll help document that and give us a feel for exactly what else is going on with this peptide in the eye. So we did a study using an atropine and a desiccation chamber to create ocular surface damage in mice and corneal nerve damage. And as you can see in the images across the top, which is the naive animal, it's got an intact subbasal nerve plexus swirl pattern. You've got the corneal epithelial terminals in the next image, and then a zoom in on those in the third. With the vehicle animal, you got obliteration of that subbasal nerve plexus and massive reduction in the epithelial terminals. Conversely, when we took those animals that had been subjected to that same damage and treated them with ST100, we got a recovery of the subbasal nerve plexus and a restoration to near naive levels on the epithelial terminals. So nerve damage, nerve recovery, another key piece of dry eye disease was happening with this peptide, and that got us very excited. Anatomical impact on an important factor in the pathophysiology. We also looked in those same animals at the lacrimal reflex synapse in the trigeminal V1 structure. We examined those tissues, and as you can see on the right-hand side of the image, the vehicle animal has a significant dropout in cell body area in V1, which is the part of the switch process for the tear ducts. Conversely, the CMP caused a significant amount of recovery in the cell body area in the trigeminal V1. We also measured something called V-GLUT2 expression, which I didn't know about until we did this study. But in any event, it kicks off and elevates when you've got significant nociceptive pain activity on the ocular surface, or if there's significant corneal nerve damage. So this provided us with an explanation for the impact on the lacrimal functional unit. Now, this data is brand new. It was about a week ago. We did a similar type of study, and we looked at mast cells, and we wanted to look at where we had degranulation of the mast cells in the lids of mice and compare that with treatment. And as you can see on the left-hand side of the image, the brighter, bigger dots are the degranulated mast cells, which are releasing histamines and inflammatory cytokines, whereas on the right-hand side of the image, the treated animals, the number of those degranulated mast cells or activated mast cells is vastly reduced. So this gives us another piece of data about the innate immune system and the innate immune response in animals and the impact that we're having on that. We'd done prior studies on inflammatory impact where we looked at the expression of inflammatory cytokines on damaged collagen substrates by epithelial cells, and then treated to repair those substrates, and saw a return back to naive levels on IL-6 and IL-8 in this particular study. And it's well established in the literature that healthy collagen is a ligand binding site for LAIR-1, which is expressed by a number of circulating inflammatory cells. Healthy collagen binds to LAIR-1, prevents the activation of those circulating inflammatory cells, Layer 1 will not bind to damaged collagen, so our repair of helical collagen structures in the eye, in particular as it relates to dry eye disease, provides us with an improvement in the expression of circulating inflammatory cells. When you combine that nerve repair, that multimode anti-inflammatory effect, et cetera, it gives us a pretty good explanation for what we saw in the controlled adverse environment chamber during our phase 2 trial. Patients on successive chamber visits at day 7, 14, and 28, these are area under the curves of their discomfort scores, and going down the chart is an improvement, by the way, saw successive improvement in their overall discomfort relative to vehicle every time they came into the chamber. So increased consistent use of the product saw beneficial effect to the patients in terms of symptoms. The protective effect grew with regular use, and we think that this gives us an opportunity to prevent recurrence of symptoms with the drug if it's regularly used. We are running our phase 3 clinical trial right now here in the US and in Australia. We've got 350 patients we're going to run through the trial, and we're very excited about the opportunity to be here today and talk to you a little bit about the drug. We think that we hit not only the nerve damage question in dry eye disease, which is important as we saw in DUCE 2, but also symptoms, issues, ocular surface tissue recovery and repair, and a broad anti-inflammatory impact on patients that suffer from dry eye disease. I really appreciate the opportunity to talk to you folks today, and if you've got questions, look me up. Thank you so much.
Thank you, Eric. Thank you so much, Eric. That's really exciting to think that there's a potential therapy that can actually reverse corneal denervation, which is really an underpinning of dry eye. So at this point, we'd love to invite up Charles Bosworth. He's the Chief Medical Officer for Azura Ophthalmics, and they are working on some very interesting technology that's targeting the resolution of MGD, and with a unique delivery mechanism and a significantly reduced dosing regimen.
Thanks, George. I wanted to thank you, George, for just the opportunity to see old friends again and make new ones. It's been a good day for that. With that, I'd like to really introduce everyone to Azura Ophthalmics. Now, Azura Ophthalmics is a biopharmaceutical company, and we're really looking at novel ocular keratolytics that target the lid margin, and then ultimately using that to treat different ocular surface conditions and diseases. So if we think about the space, we can all think about, where is hyperkeratinization present? We can think about the TFOS report on MGD. We can think about some of the reports like CLD, contact lens discomfort. And then we can kind of think about, what do keratolytics do? Right? We know that they can break down keratin, aberrant keratin production. But then there's actually this extra attribute of some keratolytics that people aren't as well aware of. And that's that some of them are actually lipogenic as well. So it kind of gives you an opportunity to attack these conditions on a multifaceted level. Now, Azura does have a compound that's actually in phase 3 for meibomian gland dysfunction. I'll go through some of the data on that today. And then we actually have contact lens discomfort kind of behind it in a phase 2 program. But we've also built a whole portfolio of keratolytic conjugates that target different things. So we have conjugates with anti-inflammatories, and you can all think about mixed disease and where that might fit. We have keratolytic immunosuppressive conjugates, and you can think about ocular graft versus host disease, among other things. But we also have ocular antibiotic conjugates, and now you can think about blepharitis. So kind of thinking about the breadth of where a mechanism could be applicable and expanding across the space in ocular surface diseases. Now, focusing on meibomian gland dysfunction, we all know since the TFOS report on MGD came out that there's a clear link between hyperkeratinization, and we also talk about protein aggregation within the meibum itself, and things like disruption of the tear film and dry eye disease. But we also have to think in obstructive MGD about blockage, dilation of the glands, and then ultimately atrophy, and then some of the diagnostics and things about how we might image that. So what I'd like to focus on today is the AZR-MD001. This is our ophthalmic selenium sulfide ointment. It actually has 3 known mechanisms of action. The first thing, it's keratostatic. So it does slow down the rate of future production of keratin through keratinocyte proliferation and blocking that. It's also a keratolytic. So when we think about protein and protein aggregation, we can think about oxygen involution into the meibomian glands and protein folding, forming these sulfur hydro bonds and groups. and we can actually break those down. And the last thing you can think about is lipogenesis. It's one of the least known attributes of selenium sulfide, but it actually helps speed up the maturation rate of ancini cells and then subsequently lipid production. So it really allows us to think about targeting things like MGD on 3 different levels. We've come through a phase 2b confirmatory efficacy study. It's what you'd expect. It's a multi-center, double-masked, randomized controlled trial. We looked at a couple of doses, a 0.5 and a 1% relative to a control. And then we looked at the efficacy and safety, of course, our primary endpoint being at 3 months in terms of signs and symptoms, but we do take our studies consistently out beyond that. So we looked out to 6 months. And this comes from the literature. When you look at a keratolytic in dermatology, they gain efficacy over exposure time. So there's really no downside for us to just go a little bit longer in the studies and see if that in fact replicates in ophthalmology. One thing that's very different about our drug is it's only dosed twice a week at bedtime. So that's very different than many of the other things that you'll hear today or you've probably thought about in terms of ophthalmology. In terms of our sign endpoint, it's related to meibomian gland pathophysiology. We look at meibomian glands yielding liquid secretion, and that's counting 15 glands in the lower lid and then looking at the number secreting liquid. In terms of our symptom endpoint, we look at OSDI, total OSDI. And I'll take you through each of those endpoints here. This is the results from that study going out through 6 months. The red box is the primary endpoint specified for FDA. And you can see at month 3, we clearly have a differentiation there from control in terms of MGYLS. And that just grows in signal until you get out to month 6. And we get about a 5-gland difference or improvement, which is about a third of scale. And you can see there's our effects that go all the way back to day 14, which was the first time point we measured. So clearly we're gaining signal over exposure time. The next thing we looked at was total OSDI. And you can see total OSDI changes here, lower is better. So we do hit a statistically significant advantage again at month 3. And that signal just grows all the way out to month 6, where you get about 9.2% change from baseline. So you can see clearly we've hit the signs and symptoms as required by the FDA in our first confirmatory efficacy study. And that led us on to some other questions, and this goes back to the clinical relevance of the effects we're seeing. So we always talk about statistical significance because that's what the FDA looks for in signs and symptoms, but we already pre-specified responder rates on each of these criteria. So the first one we looked at is MGYLS. This comes from Tomlinson's report on MGD from TFOS, and the idea of how many glands do I need to change to go from a symptomatic to an asymptomatic state. And you can see clearly we hit significant effects, going all the way back to day 14, you start to see signal. By month 3, you can see that's about 45% of the patients achieving that effect, and it gets to about 60% by month 6. So the majority of patients are having a clinically meaningful and a statistically significant effect. We can then look at the number of glands and the quality of the meibum they're secreting. So that's what meibomian gland score is. It goes from 0 to 45, and you can find the scales. They're published by Lane in 2012. You can see here, if I look at month 3, it's about 68.7% of the population responding. Now, what this means is they're getting to a normal quality of meibum, right? So they're coming in highly abnormal, some around 4 or 5, and they're getting up to well over 12, actually. And you can see if you go out to month 6, it's about 74% of the population. So now you see the majority of patients are getting open glands and normal quality of meibum. The next thing we looked at is the ocular surface disease index. And one of the reasons we like the OSDI, you go back to Schiffman's publication in 2000, and you can think about severity grading, right? And you saw it earlier, it was up on the screen. But if you get to a score from 0 to 12, you're generally classified as normal. So what we're saying is how many people get to a normal score. And what you can see, it's anywhere from 46 to 48%, or another way of thinking, roughly half the patients are getting to a normal quality or a normal symptomatic state on the OSDI. So again, quite significant effect. And you can see again growing in time as exposure continues. The next thing we looked at was tear breakup time. We do use microdosing of fluorescein in our studies where a normal cutoff is 5 seconds. But when you look at this, you can see somewhere between 2 to 2.5 seconds of gain in tear breakup time kind of consistently across our time points. So again, now you can see that patients are getting a better quality of meibum, they're getting more open glands, they're getting an improvement in tear breakup time, and they feel better. So we're finding all of our endpoints tracking together. And that leads us to the last thing. So keratolytics, they've been around in dermatology forever. We know they gain efficacy over time, and for any of you that have used them or perhaps in acne, for example, when you apply it, you might get a little tingling, burning, and stinging, but it goes away in time, right? And you can see that in our data. From 0 to 1 to 3 months, about 17% of people have a little bit of tingling when they apply it, and then in the second half of the study, it disappears. And so what we have is very mild to moderate effects that dissipate in time while the efficacy grows. Thank you.
Thank you, Charles. MGD is such a common indication here, and to have a truly effective therapy that's clinically meaningful is exciting to see on the horizon. Our next speaker here is Gary Jacobs. He's the CEO of Okyo Pharma. And Okyo is developing a drug called OK101 that has multiple mechanisms of action. That's exciting because it sort of addresses the multifactorial aspect of dry eye disease and may sort of bridge the gap to a polypharmacy approach to the treatment of dry eye.
Thank you, George, and thank you to Aura for the opportunity to speak with you this afternoon. And I have another thank you as well for Aura conducting our first in-human trial with OK101, which was excellently done. And a special shout out to you, George, for your belief and commitment and insight in helping us to understand the data that we've generated from this first in-human trial. So thank you. Most of you probably have not heard of Okyo before. We are a public company. We do trade on NASDAQ under the symbol OKYO. I will be making forward-looking statements today, so hence the safe harbor statement. We have what we believe is an exciting new drug to treat dry eye disease, what really excites us is the fast-acting and exceedingly well-tolerated nature of this drug, and also the MOA studies, the mechanism of action studies that we've done in animals that have been supported by what we're seeing in the clinic. And it's really exciting to us because what we were seeing in animals has really been supported so nicely, and I'm going to be sharing with you this first human study in OK101. So just introducing you to the drug itself, on the upper right-hand side, it's a drug that is, basically it's a lipidated peptide. The peptide part shown in the red swiggle is basically an 11-mer peptide that is key to binding to a receptor on immune cells called ChemR23. So that's a member of the G protein coupled class of receptors. That receptor is present on a variety of immune cells, but also, importantly, it's also on nerve cells, on glial cells, and that'll come to be an interesting aspect of this particular drug. We're basically only filed an IND on this in a little over a year, and this first trial was just started back in June with a randomized placebo-controlled trial. And you'll notice also that, this drug has no excipients. It's a very stable formula, a very simple formulation. We believe it's extremely well tolerated by patients. So before I do present the clinical data, I wanted to say a little bit about the animal studies. And on the left-hand side, what you're seeing is the result of a mouse model study in which we isolated the conjunctiva. And on the right-hand side of those two graphs, you're seeing the fact that this drug is potently anti-inflammatory. It reduces the influx of CD4 positive T cells into the conjunctiva. But what was particularly exciting for us was on the left-hand side, where you can see that in the conjunctiva from the mice that have this dry eye condition, you're restoring the normal level of goblet cells. For us, that was important because it was telling us that if you have a healthier conjunctiva, your naturally goblet cells secreting mucins, you could have some impact on the tear film stability. So that was exciting for us. There was another, though, element of this drug that was particularly exciting, and that was that in an animal model of neuropathic corneal pain, this was developed by Dr. Pedram Hamber at Tufts Medical Center. We were able to show that this drug, when given topically to animals that are having this severe neuropathic corneal pain, topically, it gave an excellent reduction in that neuropathic pain, similar to what you see with intraperitoneal gabapentin. And so we actually, in the next few months, will be opening a phase 2 trial in neuropathic corneal pain with this drug. That will be conducted at Tufts Medical Center. And of course, we have an IND that was cleared by FDA recently. But I want to get to the results on dry eye. So for dry eye, the trial you're seeing is a randomized placebo-controlled trial that involved a drug given at both 0.1%, 0.05%, and placebo, each cohort being 80 patients. And you can see the basically over the visits for the data that I'll be showing you. The drug itself, again, no SAEs, extremely well tolerated. We're very excited by that. I'll only be showing you the 0.05 data. This drug is amphiphilic, so it actually has both a lipid and a hydrophilic region, so it tends to form micelles at too high a concentration. So we have data at 0.1, but what we were really excited about was how nicely we saw across the board great data with 0.05, and of course FDA always likes the lowest dose with efficacy, and that's what we believe we have at 0.05. So just briefly taking you through the data itself, this is only on ITT. We are in the process of doing categorical analysis and looking at responders, subgroup analysis. We're very excited by what we're seeing in the ITT results, and we'll be following up, of course, with further data, because obviously there's a lot of data mining that we're involved with. But what you can see from this slide on conjunctival sum staining, across the board, early effect at day 15, and it was throughout the 85 days. It was supported both by monitoring lysamine green and fluorescein, so a durable response over the 85 days. In addition, we were really excited to see that even in tear film breakup time, there was an improvement in the tear film breakup time in this intent to treat. And that was again seen very quickly within day 15 and durable throughout the 85 days. And then those for the two sign indications. For symptoms, we were very pleased to see a number of symptoms where we were seeing statistical significance. First of all, in pain, where you could see by day 29 and throughout durable through 85 days a benefit. And then, of course, in blurred vision on the right-hand side and the left-hand side, burning and stinging, we saw excellent data. And I want to finish with something that was a bit of a surprise to us, because I'm sure you know that when you do daily diaries, it's a crapshoot regarding what you're likely to get. But we saw almost literally within the first 15 days statistical significance in improvement in the symptoms that patients reported in their daily diaries for both pain, burning, itching, and eye dryness. So that was really exciting to us. And we believe this drug really has legs. It has an excellent safety profile. You could see from the data on the upper hand side and upper hand portion of the slide, as well as the drop comfort results, where they were very comparable to artificial tears. So just in summary, we believe this drug has real legs. We're very excited about what we've seen in a very small first-in-human trial. It was only 240 patients, with only 80 patients per cohort. And we're clearly in the process of looking at an end-of-phase 2 meeting and looking to take this drug into registration trials. And with that, I will thank you for your attention. And again, thank Aura for the opportunity to speak.
Thank you, Gary. Wonderful overview and exciting data set in an ITT patient population. So the next guest that we'd like to invite up to the podium here is Anil Asrani. He is the CEO of Tier Solutions, and they are working a very exciting therapy called lacquer prep. And it's a restorative therapy that's trying to replace lacquer tin on the ocular surface, which is deficient in ocular surface disease. And their program has originally started looking at Sjogren's patients, and now they're also interested in dry eye patients.
Great, thank you, George. Thanks to Aura and OIS for the opportunity to present here today on Tier Solutions' approach to dry eye disease, where we focus on these 4 key tenets. And our approach really began with this question. Is it possible that there's something missing in the tear film of some dry eye patients? And our answer to that question is yes. A critical protein called lacretin may be missing, and when replaced, it has the potential to heal the ocular surface. And why are we convinced of this? Well, our founder and chief scientific officer, Dr. Gordon Laurie, a professor of ophthalmology and cell biology at the University of Virginia, dedicated his research to finding the biological basis of dry eye disease using an unbiased biochemical screen for pro-secretory factors. And that led to the discovery of a new protein that he named lacretin. Now, since then, numerous experts, as published in DEWS 2, have confirmed that lacretin is deficient in all forms of dry eye. So our thesis is that lacretin may be, for some dry eye disease, as insulin is to type 1 diabetes. If it's deficient, you have the disease and you will require chronic replacement therapy to manage it. And our lacretin-derived therapeutics, like our lead product, Lacrepep, are specifically designed to replicate lacretin's mechanism of action and therefore replace it. And that clearly differentiated MOA begins with the receptor complex, which is illustrated here, where the signaling that leads to restoration and regeneration is through NFAT and mTOR. Now, obviously, dry eye patients have stressed and or damaged epithelial cells as well as neurons, and the ability of our MOA to restore homeostasis in those cells is best illustrated here, where we used human corneal epithelial cells stressed with inflammatory cytokines that are associated with dry eye, and then treated them with either pooled normal tears or pooled dry eye tears. We monitored FOXO3, which is a marker of cellular health. Quickly, if FOXO3 is in the cytoplasm, then those cells are thriving. However, if it's in the nucleus, those cells are dying. And you can see in E here that when the stressed cells are treated with healthy tears, the cells are thriving. However, if you selectively remove lacretin from those healthy tears prior to treatment, as seen in F, they're dying. Flipping that, when these stressed cells were treated with dry eye tears in H, they're dying. But if you add lacretin to those dry eye tears prior to treatment, they thrive. And this is due to our MOA's ability to quickly stimulate autophagy in these cells, allowing them to remove damaged proteins and organelles, and thus restore a homeostatic state. Now, for the cells that have been killed or have been damaged beyond repair, the need to regenerate those cells is of critical importance. And this is data from an ARI knockout mouse that has an autoimmune phenotype, showing Lacrepep, our lead product's ability to not only regenerate epithelial cells, but also rescue the epithelial barrier function. Quickly, we're looking at days 7 and 15 in the pictures of the eyes with the following conditions from top to bottom row. You've got healthy control on the top row, the knockout plus vehicle, and then the knockout with Lacrepep on the bottom row. And you can see on day 15, the Lacrepep-treated eyes look a lot like the healthy controls from a staining perspective. And again, in the black pictures, Lacrepep-treated eyes, the tight junction protein Z01, which is essential to the structural integrity of the corneal epithelium and is labeled here in green, also looks like the healthy control, whereas the others do not. And not only are we seeing regeneration of the epithelium, we're also seeing regeneration of corneal sensory neurons by day 15 as well. Here we're looking at the same panel of conditions as before, and you can see in red the staining of GAP43, which is a marker of neural regeneration, that Lacrepep-treated corneas look like the healthy controls. And the ability to regenerate epithelium and neurons within a couple weeks is due to the mitogenic properties of our differentiated MOA. So given this and other supportive data, we're excited by the potential to reimagine the treatment of dry eye disease by building upon our first-in-human experience with Lacrepep, where 204 patients with primary Sjogren's dry eye were studied, which is a notoriously difficult patient population to treat. In that study, we had 3 arms, vehicle in gray, low dose in blue, and high dose in green. Patients were treated 3 times a day and were on drug for 4 weeks. And while we did not reach statistical significance in our primary endpoint of total corneal staining at 4 weeks, we had some great learnings from this trial. The most important one was that the doses that we used were too high. We've mapped the human data back against our dose response curve, which is bell-shaped. So the low and high doses that you'll see on the following slides are not optimal, as illustrated here in terms of where they fall in our dose response curve. They're indicated by the 22 and 44. Optimal dose is obviously lower than these doses. Another lesson was that moderate to severe patients, those with eye dryness scores greater than 60, which are shown here on the right-hand side of the slide, did better than the overall ITT group, which is on the left for reference. In the moderate to severe patients, we saw good trending and over 2 units of improvement in total staining at 2 weeks with our low dose. And staying focused on the right-hand side, low dose reached significance at both 2 weeks and 4 weeks for inferior corneal staining, and reached significance and units of improvement at 2 weeks in lissamine staining with good trending at 4. And from a symptom perspective, reached significance at 2 weeks with good trending at 4 in burning and stinging. And most importantly, we learned that our drug is safe and well tolerated, with only 3% of patients on low dose reporting any discomfort. So the work that we plan to do later this year is a phase 2 dose ranging study to test 2 lower doses in a moderate to severe general dry eye population, looking at inferior staining as the sign and burning and stinging as the symptom, where we expect to have results in the back half of next year. So to summarize, by replacing Lacretin, we're confident that we can quickly restore and regenerate damaged and dead corneal and conjunctival epithelium, as well as sensory neurons. With our clearly differentiated MOA, we aspire to reimagine not only how we treat dry eye disease, but other diseases of the ocular surface as well. Thank you very much for your time. Appreciate it.
Thank you, Anil. I think back to the patient vignette, and I think it was Ruth saying that, what was her perfect treatment for dry eye? And she said, something that's going to address my symptoms quickly, but mimic my natural tears as much as possible. So this is inspiring to think that we're replacing Lacretin to have this positive effect. So our next speaker is Riad, Sherif. He is the CEO of Oculis, and he's going to be presenting today on OCS-02, which is a potent TNF-alpha inhibitor. And he has a very interesting program that's also using genomics to identify potential responders for his therapy.
Thank you, George. Thank you, OIS and Aura, for hosting us and inviting us. Very happy to be here to present our candidate in dry eye. So Oculis is a biopharma company. We are listed. This is our safe harbor statement. Our purpose is really to drive innovation to safe side. But I would say one thing which is important to us is technology is an enabler, but we focus really on unmet medical need. And when we understand the unmet medical need, then we'll leverage technologies to address them. So what do we have in terms of portfolio? We have 3 clinical stage assets, OCS-01, OCS-02, and OCS-05. OCS-01 is the first and only eye drop product in phase 3 in DME with results which are actually at the same time point identical to what we get with anti-VEGF. OCS-02 would be the focus of the presentation, is specifically designed anti-TNF for inflammation, for ocular inflammation in development in 2 programs, dry eye and uveitis. And OCS-05 is an early stage product in a proof of concept in indication, which is acute optic neuritis. Now let's focus on dry eye. So what do we know about dry eye and unmet medical needs? So we know that, and we saw it before, and actually what we saw is actually it is growing. It is almost an endemic. So it is a big market, millions of patients. It was between 6 and 50, so enormous. Big market, mainly driven by the US today, but what is really important to see is in the end of the day, there are 2 components which are important. First, today the market is driven by anti-inflammatory drugs. 95% of the value is anti-inflammatory drugs. Why? Because actually we have symptoms, we have irritation, we have inflammation, we have pain, and patients need to address symptoms. The second is even with all the drugs we have, and we have many actually, and recently we had many drugs which is good for patients, still 87% of patients are not happy and they need something else. And then you ask yourself, so what are we missing? Is it just because the disease is heterogeneous or something else? So first, is inflammation important in dry eye or no? And actually, inflammation is important. Inflammation is a core component of dry eye, and actually is mainly inflammation which creates symptoms. Now, which type of inflammation? And when you go into the details, TNF inhibitors play a key role in dry eye inflammation, regardless if it is Sjogren or dry eye. TNF alpha are key contributors into the inflammation of ocular inflammation. And in fact, the TNF alpha Humira is approved in ocular inflammation in uveitis. So what do we have? We have specifically designed the TNF inhibitor for inflammation. The biology is more than validated in terms of inflammation. We really know that it works. It's small enough, and it is important for the eye, it's small enough which allows us to have high concentration and good ocular penetration. And we had the conversation before about trying to drive precision medicine, and we have a hypothesis here which we are validating in the current trial. So the TNF act on two components which are key components of the dry eye disease, apoptosis and inflammation. OCS02 is a potent TNF when you compare with infliximab and Humira. To reach IC50 we need less, which is important in the eye, because we need to have enough concentration to reach a target actually. So what do we know about this drug? This drug was tested 3 times, 2 times in dry eye in symptoms, 1 and uveitis. The 3 trials were positive in symptoms. I will share with you the last one, and uveitis in inflammation. And the next step is to focus on signs, which we are doing. So this is the previous trial, 134 patients. The primary endpoint was change from baseline in global ocular discomfort, and the secondary endpoint were responders, and of course the safety. So good news, the product works, is efficacious, a very positive responder, the responders into the active. In terms of safety, the product is extremely safe. No burning, no blurred vision, no redness, which is reported in current treatment. And what is really important in patients who have symptoms who go to their doctor to get rid of symptoms, it works fast. After day 14, it is statistically significant. So therefore, it works fast, it works, good responder curve, and extremely safe. But what we did further, we analyzed 8 genotypes or gene variances, 4 related to Sjogren and 4 related to the TNF response. And one of them actually related to the TNF response on the receptor of the TNF showed a very high correlation between the presence of this gene variance, which is present in the US in 19% of the population, and the response. The patient who, so we had, if you recall, we had 18% of patients who respond highly in all comers. We have 75 who respond highly, who were positive to the genotype. So what we are doing now, we are doing a phase 2b, focusing on signs, 120 patients, and we have really 2 questions. One, how OCS02 works in signs versus vehicle. But the second question also, which is important, is how this genotype positive population, which will end up, actually we already announced it, it end up to be exactly 19% of the study. How do they respond versus all comers? And this will allow us to prepare for the next phase, where we will go to all comers, or potentially we go only to 20% of the population. If we have something where it is guaranteed and it works, it's much better to have something which works in 20% than trials and error in 100%. It's good for the patient, it's good for the doctor, and it's good for the healthcare economics as well. So in conclusion, Our aim is to focus on the moderate to severe. The unmet medical need is enormous. What the doctors need and the patient need, they need something which works. It works fast and it's safe. Our product showed in symptoms that it delivers in terms of rapid onset, good efficacy, good safety, and let's see the result in the ongoing trial. Readout is planned in June this year. Thank you very much.
Thank you, Riad. As we heard from the clinicians earlier today, to have a therapy that can be targeted, this precision medicine would be so helpful and reduce the frustration and help with the economics. So very encouraging to see that. Our next speaker today is David Tierney. He's the CEO of Aramis Biosciences, and they have a therapy that's targeting interleukin-17. This is an exciting program as they've spent a lot of time really focusing in on the sweet spot of the appropriate patient that has the right balance of signs and symptoms. And they also have an approach where they are testing against a positive control, which takes a lot of courage in this field.
Thank you, George. Good afternoon, everyone, and it's a real pleasure to be here. First of all, I'd like to thank Craig and Maureen from OIS for inviting us, as well as Stu, George, and the whole team at Aura. It's a real pleasure to be here. Aramis Biosciences, many have probably not heard of us, but the company basically was founded based on the research of Dr. Reza Dana and Sunil Chauhan at Escapin's Eye Institute. Their research looked at the role of IL-17A and T helper cells, 17 being the primary effector cells in dry eye disease. Much of the early research they used actually a large macromolecule, and of course, given the fact that the drug was to be ultimately delivered as an ophthalmic solution, it was deemed really not practical. So we actually partnered with Dompe, who I'm sure you're all familiar with, and using their AI platform, we developed a small chain peptide, which is now A197. Dompe have been a great partner to work with. As George alluded to, when we were sort of discussing the early program here in consultation with our KOLs, we really recognized that since this was becoming a more crowded world, we really needed to develop a product that had a differentiated profile. What we were hearing loud and clear was, one, tolerability is an absolute must, and also then, and I think you've heard it a number of times, is onset of action. Some of the earlier data and some of the other presenters showed, the harsh reality is with dry eye, certainly with the key products that are out there, is the vast majority of patients don't tolerate the products, or certainly when they do, they don't see efficacy and they basically cycle off very quickly. So it's important that you have the right product profile. As George alluded to as well, we felt, also to differentiate ourselves, we thought would be good to have an active comparator in the program as well. And given our mechanism of action, we chose Xiidra. Just a little bit about the mechanism. Most of you are probably familiar with IL-17, but basically dryness of the ocular surface leads to epithelial cell damage, which then activates immune cells which migrate to the lymphoid tissue. There in the lymphoid tissue, they basically activate naive T cells to differentiate Th17, which produce IL-17A. These cells in turn return to the ocular surface where they exert an inflammatory effect, and they also, of course, activate other cytokines downstream, which again, exacerbates inflammation. It's also worth noting that in the lymphoid tissue as well, one of the other effects of Th17 is that actually suppresses Treg function, which again causes further problem. IL-17, while new to perhaps to dry eye, and it certainly is a well-known and clinically commercially validated target. And while directly not analogous, we actually like to point out that actually if you look at IL-17, or if you look at the development of drugs in psoriasis, where IL-17 is very well known, in many ways it actually somewhat mirrors what's happened in dry eye disease, albeit psoriasis is actually many years ahead. But if you look back in the history, psoriasis was largely treated with creams and potions, and you had steroids and cyclosporin. Then actually in 2003, you had Raptiva approved, and Raptiva has a similar mechanism of action as Xiidra as an LFA1 antagonist. But of course, the real game changers occurred with in psoriasis with the approval in 2015 of the IL-17 antagonist, specifically Cosentyx. So, and they have gone on to really transform this market. So as someone alluded to earlier, I think it was maybe Dr. Springs, could there be a blockbuster out there waiting? Maybe we can make a little time to dream and say perhaps there are some parallels here and that in fact this may come true. We recently completed our first-in-human proof of concept study in 207 patients. I'm not going to basically go into full details about the study design, but some of the key features here, given our mechanism of action, as an anti-inflammatory, we were looking at sort of patients who had an aqueous deficient or inflammatory component, and we actually did enrich the population by using an inflammatory biomarker. It was a dose-response study, so we did have a number of doses in the program, as well as, of course, we included an active comparator in terms of Xiidra. The goal of the study here, worth pointing out, is this was not powered for statistical significance because it was our first study, but the main goal initially was to confirm safety and tolerability. Of course, we wanted to establish biological activity of A197, both in signs and symptoms. We wanted to evaluate dose response. We also then wanted to compare efficacy and safety as compared to Xiidra. And then we also wanted to identify perhaps the population that ultimately we would use in future clinical studies. At a high level, the results here, it was extremely well tolerated. I think A197 had 7% in terms of treatment adverse events as compared to vehicle, 6.7%. But interesting here, we actually showed significant improvement in tolerability as compared to Xiidra. We also did demonstrate rapid onset of action and showing greatest separation compared to both vehicle and Xiidra at day 28. And, this further really stimulated us to think about actually this whole strategy of a head-to-head study showing superiority versus Xiidra. One key thing that I think, we've chatted with George about and others, and again, it's been touched upon here today, was thinking about just patient selection. So it's well recognized in dry eye disease that, demonstrating a correlation between sign and symptom is certainly challenging. There's a lot of reasons out there, but one of them is that the fact that obviously patients, especially with more severe dry eye disease, have corneal sensitivity, and this really results in them, while they may get better, they certainly may not be able to acknowledge that they're actually getting an improvement in their symptoms. So, in our program, we were trying to identify a population where we would be able to show, a correlation or improvement in both sign and symptoms. So as part of that, we actually pre-specified certain subgroup analyses, and then looking at this, we actually wanted to look at certain baseline characteristics and how they impact the outcome. So what we actually found from our study here was that patients who actually had a total CFS ranging between 8 and 12, so they were on the sort of the moderate size, were kind of the population we were looking at. But then we actually wanted to look at a population where we were actually seeing symptom scores of greater than 65. So again, more of a moderate to severe population in terms of symptoms. Looking at some of the data related to this here, again, in this population, this pre-specified population, again, where we had a total CFS less than 12 and eye dryness score greater than 60, you will see here that with A197, we saw a 0.71 change from baseline, and that represents a 27% reduction in inferior CFS at day 28. This compares to a 7% reduction for vehicle and an 8% reduction for Xiidra. As I mentioned earlier, this study was, also, we wanted to use the study to power future clinical study design. So looking here at just some future powering here, we would be actually needing, basically about 138 patients to show a statistical significant change as compared to Xiidra, and that will be 90% power. Again, looking at the same population in terms of symptom reduction, Again, at day 28 with A197, we saw a 33% reduction in eye dryness score as measured on the eye dryness VAS, as compared to 12% vehicle and 8% in terms of Xiidra. So again, looking at some powering calculations, again, relatively small numbers to actually demonstrate statistical significance. So that's really kind of the story in terms of Aramis. We have demonstrated safety and efficacy of A197 in our first human study, and we believe we actually have the basis of a differentiated profile showing improved tolerability as compared to Xiidra. We have identified a population where we think correlating improvement in signs and symptoms is possible. We also have a product which we believe has a rapid onset of action. And our next step for this program now is to take this into our first phase 2/3 pivotal study, where we will be evaluating A197 at day 28, and we will be comparing against Xiidra. For any investors out there, I'll take the opportunity to say we're in the midst of raising money, so please feel free and come and see me. Again, George, thanks so much, and I'm around if anyone has any questions. Thank you.
Thank you, David. That's very encouraging. Hearing the parallelisms that we see in drug development between ocular surface disease, dry eye, and something like psoriasis or even cancer. Once you have something that's truly effective and clinically meaningful, I think we have yet to see what these products will actually do combined with the strong prevalence. So it's encouraging to see. Thank you. And one last comment too. If you picked up on it, David's talking about doing a phase 3 program against Xiidra and a superiority approach. So that's a unique clinical regulatory pathway. So that's also of note. Okay, so our final speaker of this session is Wei-Wei Chang. She is the president and co-founder of Glia. Wei-Wei will be talking about a really interesting technology, and when we think about the future and sort of sci-fi approaches to the treatment of ocular surface disease, Wei-Wei has a treatment that actually is applied to the forehead that has an acute effect on the ocular surface. And when we first did a study for Wei-Wei, I didn't believe it. But we did a randomized placebo-controlled study, and in fact, there's an immediate effect on the ocular surface with the forehead application. So welcome, Wei-Wei.
Thank you very much for this opportunity to present our product, a treatment for ocular surface disease. We've focused on dry eye and ocular graft versus host disease. And my name is C-H-A-N-G, sorry for the air. Okay, next.
It's right here.
Oh, okay.
Press.
Oh, okay. So what we have is a novel drug delivery platform on the forehead. It's non-invasive, it's not in the eye, that signals the brain to achieve the efficacy. So it's, as I said, a novel route of forehead delivery. It activates neural signaling to reach neural targets for the treatment of diseases. We have proof of concept in our phase 2 clinical trials, a number of them, but I'll talk about the first one, and the ocular graft versus host disease. We have the immediate effect of endogenous moisture in the eye within minutes of forehead application in almost all the subjects, and with long duration of efficacy. So now this slide has many things in it, but basically at the lower left, you'll see we use a tiny dab of a gel, okay? And our dispenser, one of our first dispensers is right here, okay? So now we're into a larger one that will give you more than a month's supply, applied 2 doses per day. The active is progesterone. Our selected dose, we've tested 0.25% and 0.5%, but 1% seems to be where we want to be. Okay. The key mechanism is innervation of neurons in the spinal trigeminal nucleus. So the forehead delivery stimulates the ophthalmic nerve of the trigeminal nerve. And the animal studies have, established animal studies have recognized two regions among others. So this is a key mechanism among others that one of which is related to tear film secretion, and the other one is related to pain. And we have shown activity in both of them. So in the diagram, you'll see the forehead application going to the brainstem, in which the neurons in the spinal trigeminal nucleus crosstalks with a facial nerve. This is the one that demonstrates how the tear film is created. The facial nerve, in turn, stimulates the parasympathetically the glands to give you the effect. Okay. So the animal studies, we have different models, but here I present one where we actually show a proliferation of neurons in the rostral region that's involved with tear secretion. And so we had rats that we applied the product onto the forehead, and this is a result. I think it's after 2 hours. And this has been published. So we actually measure the number of neurons as a function of activity. So we have this new unique drug delivery platform of rapid efficacy within minutes, no side effects, no serious side effects, and basically no side effects. Pain relief has been demonstrated, and therefore improved compliance of easy application, high efficacy, no side effects, and we have a fairly long duration. In most people, greater than 12 hours of efficacy. So in terms of human studies, the first one was 2 weeks, where we didn't know what was happening. We knew that, it elicited a quick response, so we only did a 2-week study. Yeah. And in 30 minutes after application and 60 minutes after application, we were able to get a significant difference on the 1%. And then I think we also did the 0.025%. And so we further looked at different symptoms, and we were able to demonstrate sustainment of this difference from placebo on a daily basis. And I didn't show the results on staining. We did get a difference in central corneal staining. So we also conducted a study in ocular graft versus host disease patients. And here I'm only presenting, because it's a short presentation, okay, the Sandy questionnaire results and the corneal staining results. Okay. And on the Sandy questionnaire, you can see the blue, the reduction in symptoms of the active, okay, and the red is the placebo. So the placebo control phase was 10 weeks, and we had 22 subjects in the active group and 11 patients in the placebo group. The imbalance was because the investigator wanted to make sure most of her patients got treated. So that's how come we had the 2-to-1. But statistically, we were, we're actually 11 versus 11. So from a power point of view. On the right side, you'll see the different regions of staining, and I think central and inferior were the best. But right-hand side, you see the total difference. And this has also been published. Okay. So this study actually went on for 2 years. Where after 10 weeks of placebo control, you see the blue dots that represent, the blue lines that represent the placebo patients, and the red lines that represent the actively treated patients. So after 10 weeks, everybody went on to active treatment. And you can follow where the placebo patients, when they got hit with the active, that their symptoms improved. And let's see, sandy, well, it's actually staining on the left side and sandy on the right side. And the effect continues and actually improves over the 2 years. And we kept it, even though it was open label, it was pretty well, we didn't know who was in what group. Okay, so in conclusion, we have a product that has rapid efficacy, but long duration, no side effects, provides pain relief, and improved compliance. And there are many symptoms that are where we have efficacy for, including grittiness, pain, blurriness is a strong one. So anyways, it's the whole trial would be, I could spend a couple of hours here on the results. But we're very excited, and this baby boomer really wants to retire, so I really have to get this approved. I said to my partner, Ken Sawyer, I said, I do not want to run another phase 2 trial, no more. It's got to be phase 3. Thank you.
Thank you so much, Wei-Wei. Just amazing to think that you can apply something to the forehead and it can have a pronounced effect on the ocular surface, especially in patients who have severe signs and symptoms such as GVHD. So really exciting stuff. So that's the conclusion of our future therapies in dry eye and ocular surface disease. I think it's so wonderful that we've seen agents that are fast-acting, agents that are having robust effects on symptomatology, agents that are comfortable, agents that are very specialized to different underlying etiologies of dry eye. And these are many of the things that the clinicians discussed earlier today and were on their wish list. So very encouraging. Also, just keep in mind that this is a sampling of all the innovation that's happening in dry eye right now. We have many, many clients and many different approaches to treating this debilitating disease. So there's more where this came from. So thank you, everyone, for your wonderful presentations.